Hello. So I'll be speaking about a project today that was rather unusual for me. It's a translational project that mainly grew out of my clinical role here at Yale. So just for most people who know me, or if you don't, most of my work has been in biomolecular NMR and other biophysical techniques, and this is probably what justifies my activity within the development therapeutics group. So I've solved a lot of structures and done ligand binding studies with many members in pharmacology and throughout the cancer center. But today's project involves something that happened around 2010. At the time, a guy named David Pieper, who is the fourth one, the tall guy, he was a microbiology fellow in our department and wanted to learn something about mass spec. So I've been responsible for therapeutic drug monitoring within the clinical chemistry lab here at Yale Hospital and brought in lots of mass specs to do that. So we started a project in attempt to identify carbapenenase activity in clinical specimens after there was a small outbreak here at Yale Hospital. This seemed to generate some interest, and we started a small company, applied for a patent, went along quite well for a few years, and we were evicted from our space at Science Park in April 2014. And essentially, I think all I have is penny stocks from this company. But the idea was to use mass spectrometry to develop novel assays that really could have major clinical impact. And so what this first assay was designed to do was provide us a more rapid way to pick up multi-drug resistant gram-negative rods, or really carbapenenase expressing gram-negative rods with hospital-acquired infections. So I don't think I have to tell any of the clinicians in the room that this is currently a big worry. It makes MRSA look like a puppy. So there's a well-documented delay in appropriate antibiotic therapy when someone is infected by one of these MDR-GNRs, and this clearly increases patient morbidity and mortality, probably differences as much as 20 to 40% higher mortality if you have a resistant infection. Length of stay to treat the infection is between 3-4 days for a susceptible and 10 to 14 days for a resistant infection. And the hospital costs, I'll talk about it at the end, but they're significant. And I think people have talked about the fact that if we don't control these incoming multi-drug resistant gram-negative rods, medicine will jump back a century, not letting us to use chemotherapy anymore, do transplant surgery, and other procedures. So just to remind you, in gram-negative rods, there are multiple mechanisms of beta-lactam antibiotic resistance. I'm sorry I don't have a pointer. Does this work? No. So the beta-lactam antibiotics work within the periplasmic space. They have to get in through porins, so you can have modification to porins so that the antibiotic can't get in. Once it's inside, where it acts on penicillin binding proteins, beta-lactamase enzymes can break them down, and then there are efflux pumps. So generally, I'm not a microbiologist, but the way I see things is in Enterobacteriaceae, your gut organisms, they seem to rely mostly on the beta-lactamases. And non-Enterobacteriaceae always have a combination, and that makes things more complicated for us. So this is my very simplistic way of thinking about beta-lactam antibiotics. There's all the original ones on the left, and those were degraded by the original beta-lactamases that were identified and grew from our use of beta-lactams. And I'm skipping a lot of drugs in between, but basically we stand now with the extended spectrum cephalosporins, which you see listed. And this is mainline therapy at Yale Hospital for someone admitted with or someone who develops a inpatient infection. And what we have, I understand, about a 10 to 20% ESBL rate in this hospital. So those patients who, after a couple days of being on extended spectrum cephalosporin, show positivity for ESBLs or show clinical failure, they then switch to one of our carbapenems. Carbapenems are last line therapy. The other drugs after that is like colistin, fosfomycin, older antibiotics. Carbapenemases, luckily locally, are probably around 1 to 3%. It's in Europe and in Asia, rates are as high as 40 to 50% of inpatient gram-negative rod infections, and that's obviously what we want to prevent here. So this is complicated, I know, but I wanted to give you a feel for what the traditional microbiology lab workflow is. It's not my laboratory, but I think I've talked about it enough to understand it. And I divide it, separating blood cultures from urine, sputum, and fluids. What you understand is that when we receive a specimen in the clinical laboratory, first thing is done is, if it's not blood, it's plated onto agar, and it takes 12 to 24 hours, and since the lab kind of works on a daily basis, it's really 24 hours later before a colony grows, and then we begin the process of identifying the species identification and determining the antibiotic resistance. There are some rapid ways to do things, but for the most part, we still rely on century-old technology of can the bug grow in the presence of an antibiotic. That's the gold standard, and that usually takes up to 48 hours from when the sample arrives to be determined. Obviously, we need to move this timeline up to prevent outbreaks, control spread, and better treat our patients. Blood is the same, except for the fact that you draw blood cultures, and the blood cultures take 12 to 24 hours to become positive, and then we begin the same process of plating. You should know that a major breakthrough has occurred in microbiology, these MALDI-TOF microbial ID. So they work, and one is now FDA approved, the bioMérieux Vitek MS. They have this upstairs in our clinical microbiology lab. So you can take that colony as soon as it grows, put it on a slide, add the MALDI matrix. You get a protein pattern on the right, and you can, within 2 minutes, identify what the species is. Now, we can't do this for the hundreds of samples we receive per day, but it's being applied selectively. I don't know if Dave is here, the current microbiology director, but you'll see more widespread use of this. It'll eventually become high throughput. All right, so here's a little throw towards cancer. This was a case that we just made up for illustrative purposes, but the idea is if you have an inpatient with, say, leukemia who develops signs of a bloodstream infection, they'll get started on IV vancomycin for gram-positive and a third-generation cephalosporin for gram-negative. Blood cultures are drawn and sent to the lab. In the interim, if the patient is not appearing to respond, the assumption is, and after we know that its gram-negative rod has grown out of the blood culture, that it must be an ESBL-expressing bacteria, so ceftazidime-stopped ertapenem is started. But unfortunately, this happens only around 1 to 3% here at Yale Hospital. Later, it takes us another 24 hours to identify that this was a carbapenem-resistant bacteria, and we start some testing for that, but unfortunately, frequently, you see a bad outcome. So what we would like to be able to do is to move up the time of detection to either that second 8 a.m. or at the worst, that third 8 a.m. Well, 8 p.m. then 8 a.m., sorry. All right. So now just to contrast, where for MRSA, the success has been in DNA-based testing. You just do a PCR reaction. There are like 10 different FDA-cleared lab tests that we can do, and it's really a solved problem. Just a matter of whether or not you want to spend the money. The VA system a few years ago decided that they would screen every admission, every transfer, and every discharge, and they greatly reduced their MRSA carrier rate and MRSA spread within their hospitals. The problem with multi-drug resistant gram-negative rods, or these carbapenenases, is there's a large number of gene families. MRSA is really one genetic variant that can be easily detected. But multi-drug resistant gram-negative rods, there's many families, and each family has lots of members, and dependent upon just a few residue differences, the specificity varies from being a old-fashioned beta-lactamase, ESBL, or a carbapenemase. And so for that reason, molecular testing, that is DNA-based testing, has been hard to introduce. There are some panels that have begun to come on, but they still only detect the most common ones. They'll eventually become less useful as the less common ones expand. So our idea is that what we had to have was a functional test. The only known functional test for carbapenemase expression is the so-called modified Hodge test. This is what the CDC, when we had the outbreak here and we called up CDC, what are you supposed to do? This is what they recommend. And this test basically takes 3 days. So you need to have isolated your pathogen of interest. And what you do is you grow the pathogen on a lawn of susceptible E. coli. You put a ertapenem disc down and streak your pathogen. So that's 3 different patient samples streaked there. And you can see with one, there's the pathogen has allowed an ingrowth of E. coli. So that proves there's a secreted factor that breaks down the antibiotic. And this is considered the gold standard. Our idea was let's just do that in a couple hours. So we wanted to prove that the pathogen can secrete a factor that would destroy beta-lactam antibiotics. And even though there's lots of different beta-lactam antibiotics that structurally are very different, and there's also a lot of different enzymes from different enzyme families that break down the antibiotics, they all do the exact same thing. It's the Achilles heel of a beta-lactam, which is the beta-lactam ring. So you can see at the bottom that ertapenem, when it is acted on by 8-beta-lactamase, simply cleaves the beta-lactam ring and inserts a water molecule. So that is a predictable 18 Dalton increase in its molecular weight. It means that we can use the drug that has the right specificity. So the other assays that come available where you take a beta-lactam, you put a fluorescent probe onto it, and you look for a change in fluorescence when it's hydrolyzed. That works for the older beta-lactamases, but because of the specificity, you're not using the real drug. So you're not going to really match the functional specificity with that sort of assay. Another thing that we liked about this assay was that, like PCR, an enzymatic-based method will naturally amplify the detected signal. You need very, very little enzyme. All you need is enzyme and drug. And if you wait enough time, you'll eventually see enough conversions to be sensitive. So this was our assay protocol that we developed. It's about 15 minutes of hands-on time. So basically we take the colony, we put it in growth broth with ertapenem. Coming up with the appropriate growth broth was a year's worth of work, something that would optimize the activity of all the different enzymes. And then after we just incubated 37 degrees for an hour was just one time point we chose, we added acetonitrile, spin everything down, and put it onto the mass spec. So it's an easily amplified assay that we can run lots of samples on. So this is just what the liquid chromatography. So we're running on an LC-MS. We use liquid chromatography with just a 2-minute runtime. Normally chromatography, you have an effort to do 10, 20 minutes to carefully separate out your molecules, but all we're doing is just desalting and trying to purify our drugs away from anything else that could interfere. We hook it to an electrospray. So the way that you get a liquid chromatography device into a mass spectrometer requires gas phase ions. It used to be we could only do GCMS. Was this invention, which was done here at Yale, of electrospray ionization, where you can take a liquid stream and get at the other end charge, either negative or positively charged vaporized ions. Now that goes into the mass spectrometer. And all of these mass spectrometers, these triple quad mass spectrometers, they're set up similarly with 3 serial mass selectors or ion guides. And we use the first and the third, or what's called here Q1 and Q2 mass selectors, as a way to select for different ions. The one in the middle is only there to fragment molecules from their precursor molecular weight into their product molecular weights. And so when we inject, say, ertapenem, we can select for ertapenem on the first one, fragment it, and scan and see what all the product ions are. So we were able to get hydrolyzed ertapenem and the parent compound. These are the product ion spectra. So at 476 on the left for ertapenem, the highest one, that's a little bit of unfragmented parent ion, and then everything else is just fragments of that. So we chose the strongest one at 432. We did the same thing for hydrolyzed ertapenem, and you see it's just 18 Daltons increased. So now that we have this optimized, chosen precursor ion and product ion mass, we can do something called selected reaction monitoring, where we set the first quad on the desired parent molecular weight, we fragment, and then we set the second one on the desired product molecular weight, and we can actually switch the mass spec every 5 milliseconds and look for different compounds. So during a 2-minute elution, we can maybe scan for 20 to 30 different molecules just by every 5 milliseconds switching from one MRM to another. So this is what the chromatograms look like for the hydrolyzed ertapenem and the parent ertapenem MRMs. We're looking for both of them all the time. We've just drawn in blue the signal that was when it was doing ertapenem, and in red the signal when it was doing hydrolyzed ertapenem. And we just bought 2 ATC strains, one that was known to be carbapenenase expressing and one that wasn't. And this was an experiment that Dave and I did on mass specs upstairs on our own, working one evening. And what we found was this very robust hydrolysis that was complete in 40 minutes. And we were also quite impressed that the parent there was no hydrolysis overnight of the negative control, and the hydrolysis form was very stable. So that made us very optimistic. It almost would be a binary assay. In order to do a more comprehensive analysis, we hunted all over for resistant strains. First we started with, there's a -80 degree freezer upstairs where they've been collecting clinical isolates for the past 20 years. And we started going through those, and this is how we characterized them. Basically, we did all the phenotypic tests, and then we developed a bunch of PCR primers to pick up all the different isoforms of the carbapenenases, and we sequenced them using our Keck facility here to prove that it was the isoform that we were interested in. Here was some examples of those selected isolates. But what it began to show, for example, if you look at the second row, first column, is you don't always get complete hydrolysis. So NDM1, this is this New Delhi metalloprotease that came out of New Delhi and moved into Europe due to medical tourism. And it's caused quite a bit of a stir there. Anyways, it cleaves much more slowly. And so what we decided what we would do is we would distinguish positives from negatives. It's on the bottom are all negatives, by creating a hydrolysis ratio. We just took the intensity, we did an automated measurement of the areas of both peaks and took an intensity, and we wanted to find a statistically rigorous threshold that would always distinguish positive from negative. So this shows an example of our first study, just using isolates we found from Yale, which except for the NDM1, by the way, there's never been any NDM1 at Yale. All we've ever had at Yale is KPC that we could find by going through this freezer. KPC is just the most common of the carbapenenases, and it's what is most prevalent in the United States. That NDM1 was something we got from another hospital. So you can see this is the positives are at the top. On the bottom are ESBL. So these are isolates we found that expressed extended spectrum beta-lactamases. Because our concern was if we're getting a false positive, it's going to be from an ESBL. Maybe it's able to have a very slow breakdown of our desired drug, but it really wasn't at all. So then there's the areas of ertapenem, the hydrolyzed ertapenem, the intensity ratios, and for the positives, they varied over a large range. But for the negatives, it was always just background, 0.02 or 0.03. There was one E. coli that we found that does not have a KPC. We tried over and over to amplify whatever was responsible for carbapenem hydrolysis, and we could never identify the gene. But it was repeatedly positive by the modified Hodge test, and the modified Hodge test is the gold standard. So this is something that this assay will detect that no other assay would detect. That was our initial study. We published this in a journal, and we only did Enterobacteriaceae because that's mostly what we had in the freezer. Then we went to non-Enterobacteriaceae and everything fell apart. Non-Enterobacteriaceae, so the Enterobacteriaceae, they make the enzyme, they secrete it into the media. You can get rid of the bacteria and just use the media. It breaks down the drug like that. Non-Enterobacteriaceae, Pseudomonas, are smarter than that. They do not secrete anything into the media. They just make enough of the enzyme in the periplasmic space to break things down, and they usually also have porin mutations or efflux pumps to contribute to their resistant phenotype. So what we were asking the bacteria to do is have the drug get in, hydrolyzed, and out for us to get a signal. And so it took a lot of work to get that to work. These are 400 isolates that we mostly collected from other hospitals around the country. We had MTAs for all of them. We have a good selection of clinically significant bacteria, as well as all the known carbapenemase genes have been identified. So ertapenem worked terribly, and ertapenem is the most structurally complex and is one that's most susceptible to these porin mutations. It's hard to get in, it's easy to pump out. Imipenem is the least structurally complex of the imipenems, so we did both meropenem and imipenem, hoping that would solve it. The short answer is imipenem works. But so here's a bunch of data. You can see for ertapenem, meropenem, and imipenem, pretty colors, but this is the better way to summarize this data. This is what we call this receiver operator characteristic curve. And so what we have is using the thresholds are shown here on the ratios are shown in the left side curve. And then the dotted line is our optimal threshold, and the curve on the right just shows the sensitivity and specificity as we move that dotted line up and down. And what you can see is for Enterobacteriaceae, it's beautiful. But for non-fermenters, or our rapid way to determine something as a non-Enterobacteriaceae, we only have 37% sensitivity, which is pretty awful. You have to agree. Meropenem's a little bit better, and imipenem's finally is the best that we could get. We got up to 89.5% for non-fermenter sensitivity, with an overall sensitivity of 95%. So we thought this was good enough to go and start doing a clinical trial with this. And we were going to do it with Sloan Kettering. The company went under before we got our phase 2 SBIR. Everything up to here was funded by phase 1 SBIR. We do know now one trick. I went to a Gordon Research Conference and presented this work, and we had tried different ways to crack the bugs to get them to release the enzyme. But it turns out, according to the people at the pharmaceutical companies, the best way to do that is just EDTA. So if you add EDTA, it takes away all the divalent cations that are holding the outer plasma membrane together and will release the beta-lactamase. If you add lysozyme or something like that, you crack the cell and you get so much junk, you're not really able to get specifically the beta-lactamase. So if we had to do over again, I think we could get that non-fermenter up to into the 90s. All right, so everything you've seen so far has been done on an isolated bacterial colony. I just want to quickly say that we'd like to do it on other forms as well. What time is it? 12:49. Okay. Sorry, I got a phone call and I couldn't see what time it was. All right, and so we've done it on blood cultures. The people who do the MALDI-TOF ID have done us a great favor because they're very interested in not waiting for the culture to grow as well. They'd like to do it on primary specimens. So they've got a protocol where once you have a positive blood culture, they purify the bacteria out of that, and they get enough bacteria to put on the slide and identify it. So we can take the same bacteria, put it in our media, and see if it hydrolyzes antibiotics. And the answer is does, no problem. The same thing is true for urine. They've got a protocol where if you have 10 mils of urine at 100,000 colony-forming units per mil, which I understand is the threshold for a clinically significant infection, they can get enough to identify, and we can get enough to determine if it's antibiotic resistant. So this is sort of the conclusion slide, or a little bit away. At this point, we think we've got a very good assay for urine, sputum, and fluids when the bacterial colony has been isolated. We think that we can easily adapt it for urine when it first arrives. I don't know if we could do that for like joint fluid or sputum or for wounds, for example. And for blood, after the culture is positive, I think within a couple hours, we'd be able to tell you if you've got carbapenenase activity. All right, the main reason the company failed, I just want to put 2 slides on this, is the final, we got a call from the CMO of a major diagnostic company who basically said, you don't have a good answer for why my salesperson can walk into your hospital and say, buy this. Because most of the time when diagnostic companies are selling an instrument, they're replacing something else. The way that immunosuppressants got put on mass spec here is the antibody-based test was $20 an assay. We were spending $250,000 a year on that, so I could easily buy a $250,000 mass spec. But this is going to be an extra test, something that's put in and is not going to replace the gold standard growth happening 3 days later. So you need to come up with some way to financially justify it. And so the way that I've done that now, after the company closed, was develop a model. I don't have time to go into it here, but it's based on the fact that very good data has shown how much it costs for each day of delayed antibiotic therapy. And that on average is about $40,000 is increase in cost of treating an infection if you've